iron kinetics, and the lab assessment of this. So an overview of iron distribution in the body. Iron is an essential element that is critical for many physiological functions, especially for oxygen transport, enzymatic reactions, and cellular respiration. In the human body, iron exists in different compartments, and each compartment serves a specific role. So the first compartment we're going to talk about is functional iron. This compartment includes iron that is actively involved in the biological process. So the main components of this is your hemoglobin iron. This is about 65 to 70% of total body iron is found in hemoglobin within red blood cells. This is responsible for oxygen transport from lungs to tissue. Also includes myoglobin iron, which is found in your muscle cells, helps to store and facilitate oxygen delivery within the muscles. We also have iron-containing enzymes. So iron is a component of many enzymes involved in energy production, electron transport, such as cytochromes, and DNA synthesis. The next compartment is your storage. So iron is stored in tissues as a reserve for future use. This is particularly important during periods of increased demand or insufficient dietary intake. The main storage forms of iron are your ferritin. This is the primary intracellular iron storage protein. It is soluble and can release iron when needed. Then we have hemosiderin, which is an insoluble aggregate of partially degraded ferritin. This appears mainly when iron stores are excessively high. The storage iron is mainly located in liver hepatocytes and macrophages of the reticuloendothelial system, especially in the spleen and bone marrow. And then we have our last compartment, which is the transport. So iron transported in plasma bound to specific carrier proteins. The main component is transferrin. So you can remember transferrin with transport, starts the same. So transferrin is a glycoprotein that binds ferric iron tightly but reversibly. It transports iron to cells by binding to transferrin receptors. Only a small fraction, about 30% of transferrin, is saturated with iron under normal conditions, leaving that reserve capacity. Transferrin will deliver iron to developing erythroblast in the bone marrow for hemoglobin synthesis, and it will also send some to other tissues that are requiring iron for enzymatic functions. So clinical relevance for iron, we need to understand the iron compartments. This helps us to diagnose and manage disorders such as iron deficiency anemia, which is a depletion of storage and functional iron. Iron overload disorders, which is excess storage iron, also known as hemochromatosis, and chronic diseases that can affect iron transport, like altered transferrin levels. So an overview of the iron cycle regulating systemic body iron. So iron, as mentioned before, is an essential element for vital biological functions such as oxygen transport and cellular metabolism. The body tightly regulates iron levels through a complex cycle that involves absorption, transport, utilization, recycling, and storage. This regulation helps to ensure adequate supply while preventing toxicity from excess iron. Iron absorption. So iron is absorbed primarily in the duodenum, which is the first part of the small intestine. Specialized cells called enterocytes in the duodenal lining take up dietary iron. Once absorbed, iron crosses the enterocyte and enters the plasma via the portal circulation, which are the blood vessels that lead to the liver. And then we have iron transport. So in plasma, free iron binds to apotransferrin, which is a transport protein. This complex forms transferrin, which safely transports iron through the bloodstream. Transferrin delivers iron to cells that require it, especially developing red blood cells, also known as erythroblasts, in the bone marrow. Iron utilization in red blood cells, so it can help with this as well. So within erythroblast, iron is incorporated into hemoglobin, the oxygen-carrying molecule. Mature red blood cells circulate in the bloodstream, delivering oxygen to tissues. It can also do iron recycling. So when blood cells age, after about 120 days, they are phagocytized by macrophages in organs such as the spleen and liver. Macrophages degrade hemoglobin and can remove iron, which will release it back into circulation. This recycled iron binds again to transferrin and is reused by other cells, especially for new red blood cell production. This is regulated by hepcidin. So we have hepatocytes, which are liver cells, sense the body's iron levels, both stored and circulating. So when iron levels are high, Hepatocytes will produce hepcidin, which is a regulatory hormone. Hepcidin will act by inhibiting iron absorption by enterocytes in the duodenum, which will block iron release from macrophages and hepatocytes. This reduces the iron available in the plasma, which prevents overload. Hepcidin feedback and iron homeostasis. So when the body iron decreases, such as in iron deficiency or increased demand, hepcidin production will decrease. So reduced hepcidin levels allow for increased iron absorption in the gut, and it can also promote release of recycled iron from the macrophages. So this feedback loop maintains systemic iron balance. Some clinical relevance. So any disruption in this cycle can cause disease. So iron deficiency anemia is an option. So that would be an inadequate iron absorption or loss. Hemochromatosis is excessive iron absorption due to low hepcidin. Anemia of chronic disease. So this often involves elevated hepcidin, reducing iron availability. So we need to be able to understand hepcidin and iron recycling for diagnosing and managing iron-related disorders. So the regulation of iron homeostasis. So an overview of this, the body maintains iron levels within a tight range to prevent both deficiency and toxicity. The regulator of systemic iron balance is the hormone hepcidin, as mentioned earlier, and this is produced in the liver. Iron homeostasis is controlled by regulating iron absorption, recycling, and release into plasma. Response to rising body iron levels. So when the body iron levels rise, the liver will detect the increased iron through mechanisms sensing circulating and stored iron. In response, the liver upregulates the production and secretion of hepcidin into the bloodstream. Hepcidin will then travel through the blood and target cells involved in iron regulation, so mainly those enterocytes, which are the intestinal cells that can absorb dietary iron, macrophages, which are the ones that are recycling iron from aged red blood cells, and hepatocytes, which are those liver cells that are storing iron. So on these cells, hepcidin binds to ferroportin, which is an iron exporting protein, which is located in their membranes. Hepcidin binding causes internalization and degradation of ferroportin, which effectively blocks iron export. As a result, iron becomes trapped inside the enterocytes, macrophages, and hepatocytes, and this prevents iron from entering the bloodstream, leading to a decrease in plasma iron levels. So as circulating iron levels decline due to hepcidin activity, the liver will sense the lowered iron status. And consequently, the liver then reduces hepcidin production. With less hepcidin circulating, ferroportin remains active on the membranes of the iron-regulating cells, and then this allows iron to be exported back into the plasma, which will restore the systemic iron levels. So this is a regulatory loop, and it repeats continuously, and that helps to maintain the iron balance. So once again, if we have high body iron, we will have our liver increase the hepcidin level, which will then prevent ferroportin, and then it will cause a decreased plasma iron. If we have low body iron, we will have a decreased hepcidin, which will then make ferroportin active, which will then increase plasma iron. So some clinical implications of this. So if we have excess hepcidin production, it can cause iron-restricted anemia or anemia of chronic disease. You can also have deficient hepcidin production, which can lead to excessive iron absorption and overload or hereditary hemochromatosis. So targeting hepcidin or ferroportin is an emerging therapeutic approach for certain iron disorders. So iron transport, we have enterocytes, and their role is they are specialized cells that line the duodenum where dietary iron absorption occurs. They take up iron from the intestinal lumen in the ferrous form. Iron is transported across the enterocyte and exported into the bloodstream via ferroportin, which is an iron exporter protein, which is located on the basolateral membrane. Then we have hephaestin. It is a membrane-bound ferroxidase enzyme, which is expressed on the basolateral surface of enterocytes. It oxidases iron from ferrous to the ferric form immediately after that iron export. This oxidation is critical because only ferric iron can bind to transferrin in the plasma for transport. Blood. So obviously we know what blood is. It acts as a transport medium for iron throughout the body. Free iron is toxic, hence iron must be tightly bound to plasma proteins during circulation. Apotransferrin is one of those plasma proteins. It is an iron-free form of transferrin. It is present in plasma and it has a high affinity in ferric iron. The role of apotransferrin is it binds the ferric iron oxidized by hephaestin to form transferrin, which prevents free iron toxicity. And then we have transferrin, which is another one of those proteins. So transferrin is apotransferrin bound to ferric iron. Its role is to transport iron through the bloodstream to various cells and tissues. It delivers iron mainly to developing erythroid cells in the bone marrow for hemoglobin synthesis. Cells acquire iron via transferrin receptors that bind transferrin and internalize it. So iron saturation under normal conditions, transferrin is about 30 to 40% saturated, which allows it to carry additional iron when necessary. So we'll just do a quick summary of the iron transport flow. So we have dietary iron, which is absorbed by the enterocytes. It is exported via the ferroportin into the bloodstream. Hephaestin oxidizes ferric into ferrous, or ferrous into ferric, sorry about that. Ferric iron binds apotransferrin, which forms transferrin, and transferrin transports iron safely in blood to the target cells. Iron recycling. So the body can efficiently recycle iron to maintain iron homeostasis and minimize iron loss. The majority of iron is recycled from senescent or aging red blood cells. So the senescent red blood cells generally have a lifespan of about 120 days. As they age, red blood cells lose function and are targeted for removal from circulation. So macrophages that are primarily found in the spleen, but sometimes in the liver and bone marrow, can recognize and ingest these senescent red blood cells from or through phagocytosis. Within macrophages, hemoglobin from degraded red blood cells is broken down. And then the iron is released from the hemoglobin, and that is stored temporarily inside these macrophages. It is stored in a non-toxic form, which is bound to ferritin, which is an iron storage protein. This storage prevents free iron from causing oxidative damage. When needed, macrophages export iron back into the bloodstream, and this exported iron is in the ferrous form, which is transported out of the macrophages via ferroportin. Once it's in the plasma, iron is oxidized and binds to apotransferrin, forming transferrin for delivery to other cells, such as those developing erythroblasts. So the importance of iron recycling is that recycling will help conserve the majority of body iron and reduces the need for dietary iron. Approximately 20 to 25 milligrams of iron daily is recycled from red blood cells compared to only 1 to 2 milligrams absorbed from your diet. So this mechanism is essential for maintaining adequate iron supply for erythropoiesis and other cellular functions. So just a quick summary flow of the iron recycling is we have the senescent red blood cells, which are then phagocytized by the macrophages. The hemoglobin is degraded, which is where the iron is then released. Iron is stored as ferritin inside those macrophages. Iron is then exported by ferroportin into the plasma. That is where the iron will then bind to apotransferrin, which will form transferrin. And then from there, transferrin delivers iron to the cells for reuse. So in the diet, as mentioned earlier, you can get your iron from dietary sources. Of those dietary sources, one of them is red meat. Red meat is a rich source of heme iron, which is iron bound within the heme molecule. The heme iron is derived mainly from animal sources and is highly bioavailable. Another option is green leafy vegetables, and this contains the non-heme or ionic iron, which is mostly in the ferric form. This is found in plant-based foods, but it is less readily absorbed than the heme iron found in the red meat. So our daily intake of iron is generally between 10 to 20 milligrams per day. But the absorption is not great, so only 1 to 2 milligrams of iron is actually absorbed daily. The body tightly regulates iron absorption, and that is to prevent an iron overload. There are times when we need an increased requirement of iron, and that is with growing children and pregnant women. They require higher iron absorption to meet those physiological demands of, body changes and growing. Iron absorption can increase in response to these needs. Heme iron absorption. So the heme iron, which is found primarily in that red meat and other animal products, exists as iron bound within the porphyrin ring of hemoglobin and myoglobin. So some absorption characteristics: it is more efficiently absorbed than the non-heme iron found in plants. The absorption occurs intact through heme transporters in the enterocytes. It's less influenced by dietary inhibitors compared to the non-heme iron. So the non-heme or ionic iron absorption, this is mostly, as mentioned earlier, from plant sources such as vegetables, grains, and legumes or beans. It is present in ferric iron, which must be reduced to ferrous form before it can be absorbed. The absorption is more affected by dietary factors such as phytates, polyphenols, and calcium. And these are some laboratory assays that can be performed to assess the body iron status. So make sure you take a look at these and know what they are testing for in the diagnostic use.